Hello IB biology class. Today we're going to be having a look at how we compare gene sequences between different organisms and what these gene sequences might have in common and how these gene sequences might be different. Before we get into how do we do that though, we're going to cover a little bit of background information. So in 2003 they finalized the Human Genome Project. This was a monumental project. It took 13 years and cost nearly I think it actually cost over $1 billion in order to complete, and relied heavily on international collaboration among research faculties around the world. And it was done so that we could figure out the correct sequences of A, T, C, and G within the human genome. And when you know those sequences, you can start to then identify individual genes. And then one of the questions that we were looking at was, how do individual humans differ? We share so much of our DNA in common, and yet to look at two individuals, we can see many differences. And so the Human Genome Project really started to provide the answer to some of those questions. But as I said, it took 13 years and international collaboration. It was huge. And yet since that time, technology has improved exponentially, and it's improved incredibly quickly, so that an equivalent project today, if we were to sequence the human genome again today, it'd only cost us around 5,000 Australian dollars and would take only 1 to 2 days to complete. That's mind-blowing. So with the improvement in that technology, it now means we're able to sequence the genomes of thousands of other organisms. And with that information comes our ability then to compare genes across different organisms. So for instance, we can have a look at, how does a human gene compare to the equivalent gene of a chimpanzee, for instance. So today's little activity that I've got for you, this is a DNA sequence alignment lab, and it's exactly what we're going to be doing. We're going to be pulling down the gene sequence of different organisms and then uploading them into a program that will look for similarities and differences. So you hopefully have this handout in front of you. And this activity is going to use information that is freely available to scientists around the world. So we're going to be using two different databases. The first here is the GeneBank database. And the GeneBank database allows scientists who have sequenced genes from different organisms to upload those gene sequences. The second database that we're going to use is Clustral Omega, and that's more a program than a database, where it will actually line up different DNA sequences for us and help us to determine any similarities or differences in those DNA sequences. So the gene that we're going to be focusing on is called the cytochrome oxidase 1 gene. Now this gene is actually involved in cellular respiration. So any eukaryotic organism that contains mitochondria and undergoes aerobic cellular respiration should have a version of this gene. So it's a really common gene to use when we're trying to compare genetic sequences among organisms. So the first thing that we need to do, we're going to log on to the National Centre for Biotechnology Information database. That's the GeneBank. And I'm going to switch screens again in a moment, but if I just scroll down a little bit here, you can see the web address listed just up here, and we're going to launch into that page. So if I just switch us across, hopefully you've logged into this page. If not, pause the video and log in. These online databases are absolutely amazing. They contain a wealth of information, but they can be incredibly daunting to navigate around. So I'm just going to walk you through doing that, and we'll see how we go. So the first thing we need to do is actually tell the website that we're interested in finding gene sequences. So if we come up here to the search bar, instead of searching all the available databases, because there's a lot here, some of them just contain books, but some of them contain protein sequences for us. We've got short nucleotide polymorphisms there as well, but no, we're only interested in DNA sequences. So if we scroll around, here we go, we're after our gene sequences. So we just click gene, and then up in the search bar we're interested in that cytochrome oxidase gene, which is given the name or the code COX1. Alright, so having entered that in, we're going to hit search. And the database is now going to mine all the available information and come up with any sequences that scientists have uploaded. So we can see now here on the page that we've got, look at this, 1 of 20 showing of 11,444 available sequences. Now some of those sequences will be double up. So some scientists will be looking at a similar organism or the same organism and unknowingly upload the same sequence for the same organism. But all the same, it gives you a little bit of an idea of the sheer volume of information available on these databases. And so here we are, we've got all these COX-1 genes here. And if we have a look at the descriptions here, the first one is for the fruit fly. We've got Saccharomyces cerevisiae down here, that is bread yeast. If we keep going, we've got Neanderthal genetic information here for the COX-1. That would have been derived from fossils that have been found. We've got mice, cattle, this little guy down here, C. elegans, that is a roundworm, delightful chimpanzee. Okay, so your job is to go and find the gene sequences for COX-1 for 5 different organisms. I'm going to walk you through this as quickly as I can. We'll just focus on the top one here for fruit fly. You need to click on the gene ID here, so COX-1. And it should load. There we go. So now we've got a really interesting page. It gives us some of the official information about that gene. It gives you the classification of the organism here. So we know that the fruit fly, it's a eukaryotic organism. Look at this. It's an arthropod. It belongs to the insect family. And it's got various other classifications along there. Not really interested in that. We're going to scroll down. Some really fancy information here that we just skip over. And we're coming down to this region called genomic region, transcripts and products. And what I want to know is what is the gene sequence for COX-1 with our fruit fly. So I want to know the order of the A, T, C's and G's. Now to find that, I'm going to move my cursor over this little red bar here and it's going to load some information for me. Part of this information is where can we find the COX-1 gene. So we know it's a mitochondrial gene there. Look at that. It is 1,536 nucleotides long. So it actually tells us how long this gene is. And all of that's very interesting and there's some more technical information there. But if we scroll down, we're after this faster view. Okay. So if you click on the faster view link there, it's going to load us up onto another page. Wait for it. And there it is. There is the A, T, C, G sequence for the COX-1 gene of fruit flies. Mind-blowing. So some research has gone out there, he's been researching fruit flies, he's managed to extract the DNA and sequence the DNA and uploaded that information onto the gene bank. What I would like you to do with this information is highlight it, copy it, and then paste it over into a Word document. Voila. Nice and easy. You're going to need to do that for 5 different organisms. Now, I'm not going to go through each individual one here for you. You follow exactly what I've just done to get to this point. Now, when you come back into your Word document here, we've got a lovely long name up here. Oh, that's boring. Let's abbreviate that and get that much smaller. So it's really important that you keep the little symbol at the front there, and we're just going to call this fruit fly. Nice and easy. All right. As I said, you're going to do that for 5 organisms, remembering to abbreviate the name each time, and then we'll move on to the next part of the activity.